So for this part on Lab 2 of ST2 hypothesis testing, we're going to focus on looking at power, effect sizes, and biological significance. So please see the introductory videos for more. This demonstration video is more going to focus on what we're thinking about when we're looking at these things with RStudio. So what we're thinking about is how we can apply RStudio to do things like this, where we can simulate 100,000 t-tests where there's no difference between two different groups. So you can take vectors of numbers and compare them to each other where the underlying distributions show no difference. And we do that with the R-norm function, where we expect that the t-distribution we get at the end will be centered on a zero, but we will note that there'll be some tests where we hit extreme negative and positive t-test values, and correspondingly, those will tend to have small p-values. However, there's no difference. So these sort of simulations are things that we can apply with our own data when we're testing it further. You may have experimental data with particular labels, and you can do certain things like sample label permutations where you can mix the groups together, where you essentially remove all the pre-existing labels and repeat those tests between the different groups without those initial labels and see what distribution of t-test and p-values you get from that alone. And that's to identify data artifacts that might bias your interpretation of the initial labels. So you can repeat those processes, and that's something we'll look at later in other courses. What we expect for this when there's no difference is a uniform p-value distribution. So if you're testing lots and lots of genes and you're doing transcriptome analysis to look for differential gene expression changes, you would typically expect to have no differences. So if you plot your p-value distribution, you'd expect something like this. If we repeated this test, and in this instance here, had some difference between the different groups, we'd expect that the shape of the t distribution would be the same. It's just shifted in one direction to be positive or negative and centered on that value, but the shape will be similar. So find instances where we find no difference and instances where we find a large difference when the average actual difference is somewhere in the middle. At that instance, we'll find more of our results have a smaller p-value, but there are ones where we have low power to detect two effects, and this is with n equals 16. So then let's look at the RStudio to further explore how we can apply this with the package PWR there. So what we're going to do for this part on power, effect size, and sample size is that we're going to install the package PWR. So we're going to run this line 108 and install that there. And we have ask equals false so that R doesn't bug us with annoying questions. So let's activate the library in 109. And then we're going to look at this function pwr.t2n.test. If you look at the PWR package and see what's inside there, so where's PWR? You'll see that actually does a huge variety of things. And there's enormous amount of different options. We're just looking at one example function, P2N test, which is testing power for a comparison of different pairs of groups. But you can do similar stuff with ANOVA, chi-square testing, binomial distributions, and other variations on this. So we're just going to look at the help for this test. So you can see here, we have a few parameters. Firstly, you have the number of observations in the first sample, the second sample, because we're comparing groups, the effect size, D, the sig level, which is alpha, the type 1 error chance, power, the 1 minus the type 2 error chance, and then the alternative, which will generally be two-sided for us because we may not know if our data is higher or lower, which group is higher or lower in advance. And there's examples down here if you're unsure how to try it out. So what we need to consider is that this function essentially estimates the fifth parameter if we put in 4 arguments, 4 of these 5 arguments. So N1, N2, D, sig level, and power. So what we can try here is an example here, where we're putting in N1 equals 30, N2 equals 30, D equals 0.5, and the significance level is 0.05, which means it will estimate the power. So that will give us a response on our power. Here you can see that's estimated as 48%. I note that we're putting the alternative hypothesis as being two-sided. So we're effectively having the two-tailed estimate for these different features. So we can change this function and change the power if we have a more smaller alpha value. So if we have a smaller alpha value, as we can expect, our power will be smaller. If we change our D effect size to be bigger, we can expect that this power will now be bigger, which you see 88%. So if we've biologically really different groups, that makes it easier to detect them. If our biological effect is very, very small, then our power here is now 1.6%, which is very small. If we increase our sample sizes to something that's much, much bigger, we know that this power will increase, and now it's 9%. But if we had an effect size of, let's say, D equals 0.5, that would mean they're actually a really good power to detect those effects. So you can see if we change the D value, our capacity to detect effects of a particular magnitude changes. So this is where we can kind of look at the associations between these different features to estimate what we need as required. So we can change this one here and have power equals let's say 0.8 for an effect size of 0.5 with 30 and 30, 31. So what you see here is that if we don't put in the sig level when that's not clearly stated, we'll actually find that that causes an issue. So that's why we need to put in here null, so that it's reminded to test for that parameter. So see there we have the significance levels now 27%, which is going to give us a lot of false positives. So what we can do there is compromise, and if we decrease the power to 0.5, we can see the significance level decreases. So there's a sort of balance between the power we have to detect true positives versus our false positives, and as we know, we can estimate that based on using the PVV. So what we can do as well is see that if we increase D to 0.8, our alpha value will decrease, and that will mean that we can afford to increase our power. Now our power is 0.8 and alpha is 0.027. This other example here is estimating D. So if we change this, so let's put alpha to be 0.02 and our power is 0.8. I remove D with our sample size there. You can see that the effect size we would need to detect with a power of 80% and an alpha value set to 0.02 for samples of 31 and 30 will be 0.83. So that's quite a big effect. If we adjust this, so relax alpha and also the power. So remember, the power will be a function of how different the groups are. Now we should see D to be smaller. So 0.58%, 0.58 is our effect size. And again, as you can expect, if we increase our group size to 131 and 130, our minimum effect size we can detect reliably, given that power and alpha is now 0.28. Finally, we can add in D and estimate the other one of the N1 and 2 sample sizes that we need. So let's say N1 is 10 and D is 0.5, given a power of 0.6 and a significance level of 0.05. What do we get? We have a insufficient numbers for that. So N1 is too small, let alone N2. So we can't estimate N2 because N1 is way too small. So for those features, we actually need a lot of samples. Yeah, so that's why if N1 is only 20, we need 1,059 other samples. So let's increase N2. And what we would see here is that if we increase N1 by only 80, our additional requirement for samples in N2 is now just 25. So it's sort of a balance. We want an equal number of samples in each group to optimize how many samples we need to collect. So let's say if N1 is 40, you can see we have 40 and 40.3 required. So we need 40 and 41 samples in each group then to satisfy these criteria. And you can see there the power is kind of still quite small. So if we change our power to be higher, given this realistic biological effect size of 0.5, we see we have 47. So maybe we can change this to 50. Not quite, no. Let's try 60, nearly. 64. Yeah, 64. Each group can work. So you can see there a realistic biological effect size of 0.5 with an alpha value of 0.05, a power of 80%, so an 80% chance of actually detecting effects of this magnitude requires sample sizes of 64 and 64. So this is useful for planning experiments to seeing how big is your biological effect that you want to detect that actually makes a difference in the context of your experiment, and what, given alpha and 1 minus beta, what kind of biological effect size is associated with different sample sizes. You can see there the balance in the sample sizes is quite important. So that kind of concludes this this video on power testing with RStudio.